Well, thank you so much. I'm delighted to be here, and thank you for coming with the rain, which it is heavy tonight. So, you probably are surprised of the title, but I hope I'll first give you an overview of what are the things. Oh, here's my collaborators. I have to thank them first. This is people in my group that did some theoretical work and simulations. And I work with another group of experimentalists, Samuel Stoop, which is a chemist that does some of the synthesis. And this student was joined between them, he and I supervising. And also with a person that does characterization, X-ray, Mike Betsick and his group. And the funding I get is from this work was from the Department of Defense through the National Security Science and Engineering Faculty Fellowship, which was very generous, the National Science Foundation, and the Department of Energy. So I'm going to tell you, this is my group. So we do theory. Let me see the pointer, yes. We do theoretical work, and it's very mixed. They come from many different countries, the US and so on, and he was missing that day. They mainly we have people from physics, applied physics, chemical and biological engineering, and also from chemistry. So it's very diverse. And so we can attack a broad number of problems. And some of the problems I work on are summarized here. So just to tell you what sort of research is that we can assemble. So all of that involve ions. So when my award was, for example, in electrostatics, it means I work in charges, but they are not electrons, just ionic structures, like a sodium chloride that decomposes in positive and negative ions, and in molecules that are complex that interact with these ionic structures. So for what we do, for example, in this case, we do some fibers that mimic some biological tissue, like that are in the cells. So I used to make three-dimensional cells with my colleague Sam Stoop, and then he can grow cells on it and so on. So these are gels of water and a lot of ions on it, and molecules that assemble because they have parts that are hydrophobic, means they hate the water, and some parts that are hydrophilic that they want to expose to the water. So we design the molecules, or chemists can do those molecules to assemble in particular morphologies. So there are some applications. We also try to do things that are related to materials, and but made of these cases of gold with DNA functionalized aside. So to use the hybridization of DNA that they form the double helix by hybridizing some base pairs, you can sort of assemble structures like single crystals, very large. But instead of being made of atoms, they are made of particles that have DNA surrounding it. And so, you can direct the assembly. And then, of course, the gold core gives us some properties desirable for photonic applications and so on. So, you see, it's very varied. Another example of things that you do with this sort of research we do is recover nuclear waste or rare earths that they are dispersed in water. So people design specific molecules that recognize, say, europium or something to reuse, and take it out from the water and bring it to the oil, because the tails hate the water. And so they go to the oil and then they wash it out and they can reuse it. And so we sort of look at the properties of that. And they introduced me as winning an award for polymer physics, and that was in the area of polymer electrolytes, and I'll show you some examples of the application. This is the ones of the polyhedral structures I will talk mainly tonight. But I wanted to give you an introduction of the use of all of these soft matter or soft materials that we call. So contrary to for years we have been talking about solid materials, these materials that are polymeric are soft and deformable, and so they have a lot of functionality because they are not they are not covered with many bonds, but a polymer will typically be made of many chains that are in linear arrangement only. So it's very long chains. So the interactions with the neighbors are very soft. Okay? So you can actually deform them like a silly putty. And when you have charges on them, you can use them as the electrolyte that is used in batteries. And so why would you like to do that? Why would you use a polymer for, say, a lithium battery. And first of all, they are flexible, they are lightweight, they are low cost, they are recyclable, and they are non-flammable. So you have heard this Samsung, the batteries explode because when you have a change of pressure, for example, you can evaporate in a gas phase the lithium, and in that condition, it's explosive. But a polymer, where a polymer is so long, so the molecules are entangled, so they don't form a gas phase. You cannot have a huge polymer coming out and form a gas phase. So it just keeps it, safe and so on. So there are sort of like some of the application of these soft materials. Another ones are in, you can make even cables with these polymer electrolytes. If it's a gel made of charged chains, like an example of the most densely charged polymer will be DNA, right? It's a very long chain with a lot of charge. So when you have charge in nature, you cannot have excess charge in a bulk material, right? It has to be electroneutral, so it has to be compensated with the counterions associated to that charge. So if you make a gel, it has to have a lot of ions, and now if you put a potential that drives the ions, they accumulate a lot in a plate, and that's called the kind of the storage energy you can do, which is huge. We call them supercapacitors. So they are really futuristic materials, but we still don't know how to use them. They are slower, but they have other properties, like, as I said, supercapacitant. Here's an example where you can actually use it as a cable and connect it and stretch it by 100% like that, and the music still plays going through it. Okay, because it's supercapacitant, so you're just making a signal and it's transmitting. So there are many, many applications, diodes and so on. The other applications are the absorbance, for example. This is a polymer can make a gel that is flexible. Right? So it's cross-linked. So when you take a polymer and you cross-link it, it's one molecule. It's only one molecule, like a solid, but it can stretch and swell a lot. So if it's charged, it will swell because the ions have to cancel the charge, so they want to be free. So you absorb so much water, so the ions can be around, like in a diaper, you can absorb a lot of water. Yes, because it's made of polyelectrolyte gels, right? So, and then you can play with these interactions, like if you, instead of using monovalent ions like sodium chloride or whatever other salt to swell it, you use molecules that have higher valency, like 2+ charge instead of 1+, to cancel the charge, then they can start shrinking. So they can change the shapes, orders of magnitude, by what you put around them. So they are like actuators, right? They can change sizes and so on. It's slower, but they can serve to sense things and change enormously. So, and some of the properties are related to the properties of chromosomes, which is DNA, which is linked by proteins, as opposed to cross-linked permanently, but the proteins cannot disassemble, but over some length scales, it's like frozen, and so on. So they have properties of living matter. So what I'm going to talk today is that these polyhedral geometries are shells that the ones I want to talk about, which is membranes that close to encapsulate something inside. And the examples are the viruses, right? The viruses, there are some capsids that encapsulate DNA or RNA to protect it and deliver it. And then, of course, then in the cell, they can't replicate and so on, separate. But some of the things that I decided to talk about tonight is about the geometry aspects of it. These viruses, for years, people know that when they grow very large, they facet and they form like icosahedron shape. So icosahedral shapes are fascinating, very geometrical structures. There are many, many of these shells in the living matter. Some, we call them organelles, for example, some microcompartments. So in the cell, reactions take place. And if you have molecules, if you have these little microreactors, you can concentrate the materials to make the reaction, because it will be impossible in a cell to find two reactants and make a product, right? So these are like microreactors that, act like concentrating the material or catalytic. And it's huge. They are all over. And so the point is that they have this polyhedral shape, and in this case, it's the icosahedron. So you say, what is special about an icosahedron? And so this is the question that we started looking at. There are other shapes, and why icosahedron? And there are some animals that make other shapes. These shapes, icosahedra is a very magical geometric object because it's perfect. And I'll show you the type of polyhedra is called a Platonic solid, and there are only 5 of them, and I'll show you the properties. But there are some animals that also make a Platonic solid. This is a dodecahedra. The dodecahedra has 12 faces and 20 vertices, and the icosahedra has the opposite, has 20 faces and 12 vertices. And that's a very important thing on symmetry. They are made of the same shape. It's a polygon, here is a triangle, and these are pentagons. And you can only make 5 of these perfect shapes with different geometries, that all of the vertices are equivalent. And those are the 5 Platonic solids. So it's a tetrahedra. This is a cube, octahedra, dodecahedra, and icosahedra. So these are have fascinated artists, scientists for many, many, many centuries, because they are, as I told you, they are perfect geometric shapes that every face is equivalent, and the vertices the same, and the vertices are equivalent. And so even since Kepler thought that the motion of the planets had to follow this path of perfect Platonic solids. And even before that, Plato had the 5 elements. He needed 5 because he needed to have the 5 Platonic solids associated with them. So the one that was missing was the cosmos, the dodecahedron. He associated to the other geometric shapes. And so I have in a story that this paper, I don't know anymore where it went to, but at some point some scientists thought that when the Big Bang exploded, the radiation had the symmetry of an icosahedra, actually, within fully spaces of dodecahedra. So, but they do exist. These do exist, the Platonic solids. They don't exist in the universe, as they think, and Plato had to imagine the elements be associated with them. They exist in, for example, the viruses, as I said, but also the fullerenes, which are made of carbons groups. So the questions are, what are the other shapes? So, where we have capsides that take other shapes. These are called Archimedean solids. These are not so regular as the Platonic solids. These have, every vertex is equivalent, but they are made of 2 shapes. And so when you have crystals, and they are single crystals, they have shapes similar to them. It's truncated. They look like truncated Platonic solids, right? Because they minimize energy when they're a single crystal like that. And there are tons of other type of polyhedra. Sometimes irregular, these ones are called Johnson solids. There are like 92 of them. The mathematicians have worked because these objects have specific rotations, have certain symmetries, and sort of like they are able to describe them mathematically. These are more complicated because they have what is said a different symmetry, that perfect symmetries of the Platonic solids and so on. And these are n-icosahedrons. So I'm going to show you, these are like tessellations of like the moon that come together. So all of these shapes, I'm going to show you that we do find them in nature also. And there are some irregular polyhedron. And I will try to explain you why they form and whether they have some specific function. Okay, so that's the motivation of the talk. Why are molecular shells important first? I'll show you some reasons why I care about studying these. And the molecular shell geometries are the other than icosahedron. I'll show you yes, they are at the level of molecular level forming. And why are they buckle? And why will they form less symmetric and possibly non-homogeneous shells? Like here in this example I showed before, I showed that this polyhedra has 2 different colors, like non-homogeneous in composition. So these are some of the things I had been asking and to construct. So I would say why I'm interested on it. First of all, because there are very many biological examples, as I told you, of these polyhedral shells in the viruses, organelles, and other what are called archaea organisms. Archaea organisms are organisms that live or can function at very high concentration of salts, like the Dead Sea. Okay? Some bacteria also can resist a lot of salt. You know, the human cells generally die when you add a lot of salt, but some bacteria can support a lot of salt. So they can live in harsh conditions if you want. So these carboxysomes, the one I showed you before, these are particularly interesting for energy too, because they contain enzymes that they are involved in photosynthesis. They concentrate carbon dioxide to convert it into sugars and energy and so on for the cell. So there are some examples here where the textbook says it's an icosahedra, but the fact is that the new images show that they are not really icosahedra, that these shapes are not corresponding to the perfect Platonic solids, but more other type of irregular polyhedra shells. And so you make projections of them and you realize that it could not fit the shape of a polyhedra. So they used to call them quasi-icosahedral, but I will explain that there are other shapes that arise. So they're halophilic organisms, are like the archaea organisms that support high salt concentrations. They also have these sort of geometric shapes like triangles or these sort of squares. And they used to call them square bacteria or triangular bacteria. They are not bacteria because the cells are very different than the bacteria ones. But they are called archaea organisms or halophilic organisms. Archaea organism is the name for other type of the origin of life, that the first microcompartments. To have life, you have to bring components into a compartment. So cells, the same cell is a microreactor, right, with many components. So you have to concentrate them to react and make something. So they're associated with some sort of the origin of life organisms, but not bacteria. So as I said, I'm going to tell you why it's important. So I'm going to describe first the membranes. The membranes, actually, they are covered, for example, the cells. They can be membranes of mammalian cells or some more primitive cells that have, in bacteria and so on. So they generally are made of these molecules that have tails that hate the water. They're called amphiphiles, and heads that love the water. And then they hide here the hydrophobic parts, and they expose these. And they can, these amphiphiles, if they have two tails, they generally form bilayers. And these bilayers can close and make sort of a shell with some water inside. And if it's small like that, it's called a vesicle, but they can be very large too. When they are very large and it's a cell membrane, then you can think of it as flat almost, right? Because the curvature is very big. So they can regulate a lot of the processes in the cell, going between one extra internal to the cell or external, the ions that go. If they have only one tail, they generally don't form bilayers. They form micelles only, where they expose the head to the water and they hide the tails. So these are some of the morphologies you find in what we call the liquid vesicles and bilayers. So this is an example of, is it playing? Yes. This one is not playing. So there are two types, for example, how they're useful. These bilayers that form for the cells, they can permeate proteins or even ions, making these channels, side channels, and so on specific. And they're all the way to permeate through the membranes. The water goes through the bilayers and can pass to one side very not easily, but when it's liquid, they go through them. And as I said, in water, they can regulate the concentration of ions inside and outside. And so some materials, where they are very small, they actually want to go through the membrane, and so they have a positive addition to go there. There's some drugs and can be taken, but can have a barrier to go through the membrane of the cell. And the ions are passive. They just have a huge barrier to go, because the ions have the water around surrounding them, so you have to make a special protein to make the ion channels and so on. But water passes easily from one side to the other. It passes easily when it's liquid. So it happens that when you have a room temperature, this is in Kelvin, so it's like room temperature, these bilayers are kind of liquids with the two tails. And this is an example of a very heavily studied one that is associated with pulmonary cells and so on. So it's liquid, so the water can go easily. And then when you lower the temperature, they form what is called the gel phase. A gel, as I tell you, is like it's like crystalline, because now it has elasticity as opposed to be liquid that they move around. So they have a transition. And so this transition can be also induced when you add multivalent salts, like divalent salts or trivalent salts. Then you crystallize these structures. Okay? So when you crystallize, they have different properties. Now the water change, but the evaporation of water is different. So for example, in the archaea organisms that are more rigid, is there less permeation than in the ones that are, the cell membranes that are more liquid at room temperature. So this just summarizes that, in order for the water to go, this has this passive permeation, you need to have a barrier to go in energy, because the water likes to be in the water region, so it has to go to this hydrophobic region. But this barrier, for example, is different if it's, this barrier will be for when it's in the liquid state and it's in the gel phase. And in the gel phase, not only the barrier is slightly larger, but it has less fluctuations. And so that happens for the two examples I show you. So basically, the solid or the gel structure has less permeation of ions. So this is some of the conclusion of this is like fluctuations allow you to go to the barriers. By fluctuating, the ions can go through it. And the fluctuations of the bilayer are larger than the one that is crystalline or gel. So why do I care about that? Because I'm going to show you that these buccal shapes and the ones that you deliver into cells, like with the virus and so on, are crystalline. And when they are crystalline, they have different properties, and they have different properties of formation and so on. So the driving force, when you have something that is crystalline, we call it elastic. It's an elastic membrane. Is the one I'm referring to the gel phase or a crystalline phase. Besides these sort of driving forces for the assembly, you have the geometry. They take these closed shells that I'm going to describe of the membranes, depend on some other parameters that are mechanic parameters. Like one is the Young modulus. You choose the ease at what you can compress something on a stretch. So an elastic material, you stretch it and you let go and it will return back. If you have a liquid, it would flow, right? So it doesn't have a Young modulus. Membranes that are liquid can actually bend and have an ease to bend. And so they do have a bending modulus. So that we characterize the membranes into two types, the ones that are liquid, fluid, and the ones that are crystalline. So the ones that are fluid, I show you some examples, do not have a Young modulus, and these ones have a Young modulus. And the ones that have a finite Young modulus are the ones that, when they are small enough, can facet into these shapes, into these polyhedral shapes. So I hope just to show you with this example that it's important to know whether some membrane is crystalline or not, and that the shape is associated a little bit of the properties it will have, in terms of passing or allowing the permeation of water or other components. So in the examples of the design I'm going to do, I'm going to show you how when you coassemble a positive and a negative amphipyle, this is the tail that has the water, and this has -1 and +3, you form very anisotropic shapes. And if you remember, this looks very much like the shape I show you of the bilayer. But then when it closes, these have regions where it's softer. So I'm just trying to show you that these polyhedral shapes that you might see, this is a projection cut of something closed, can have different properties, different areas. So it can permeate different in the same vertices or the folds and so on. So that's what I was interested in studying, these possible polyhedral shapes. So what we use is to make them is this assembly by ionic, bringing a positively and a negatively charge. But what it keeps them strong is really the tails. I'm going to show you that when they come together, they crystallize the shell and they can form very strong bilayers. Okay? So this is what the people use to assemble things in my field, electrostatics for. Electrostatics is used to bring the things together that otherwise will not mix. But then, you have to make sure you have other mechanisms of reinforcing that structure. And so in this case is the hydrophobicity of the tails that was, when you bring them together, it's going to crystallize them and make them very strong. Okay, so this is an example of what we call a coassembly of two components that, if they were not charged, they will not come together. Okay, because this head group is very big, and the tails, you don't know how to pack. There is no reason why they will form an assembly together unless you have some mechanism to have them attract to each other and co-assemble them. Okay? So this is an example of a simulation. So we work with simulations. So this is full atom, has all the water inside, and every carbon, and on the tail and the head and the fraction of charges that you have. This is very difficult to do in a big system. We just do a very tiny system, and then we use different techniques to look at larger and larger scale. So we go from the full atom. Instead, the previous one I show you, this is called a Martini model. It's coarse grain because now 4 waters is 1 unit. So you make the units bigger so that you can actually look at higher magnitude things go past. Okay, we don't have the capacity now to simulate with full atom all the waters and everything, the whole process. So this is how the scientists work in my group. We have people that does full atom, coarse grain, and I'm going to show you the mechanics model. The mechanics model is to make more continuum. So this is in a new science that we call that mesoscale. You do all the length scales with different techniques, and to get an idea of what is the process and so on. So I go back, and before I go and show you how we find these buckle shapes and all that, and the physics of it, I'm going to show you the physics of why, in principle, or has been for many years, the icosahedron, a ubiquitous shape in biology. And this is only related to some pure geometry, okay? So if you're going to think of the tails or whatever molecules that are making these capsides here of being packed crystalline structures that are the most close packed. So in 2 dimensions, because they are thin, very thin, so the close packed structure in 2 dimensions is something made of triangles, where each particle can have, this particle can have 6 neighbors, these little bonds, okay? So it's made, if you look at it, it's made of triangles that make hexagons with 1 in the center. So this is the close packed structure in 2 dimensions. That's the most dense. So if you put sort of like a head group in this point, they touch and, close and the space. Because they don't want, see, if they were the amphibians, they don't want the water to go through, right? So they close pack. So what happens if you want to make a shell out of this close packed structure? So you can imagine what can happen. It's like if you're trying to pack a soccer ball or you can't. Right? It will spike, it will buckle. Because in order to actually tile something with a crystalline structure that is curved enclosed in a sphere, you have to have something that's called defects. And these defects are pentagons. They are little points. They are little points where instead of 6 nearest neighbors, you have 5. Okay? And so by geometry, this is a theorem, of geometry, is everything that is closed like that have to have, if you're going to be able to close it and be periodic, you have to have defects. And the minimum number is you have to have the number of pentagons minus the number of heptagons that has 7 nearest neighbors have to be equal 12, 1 minus the number of handles the geometric object has. So a sphere has no handles, so it has G0. So the minimum number is to have 12 defects that are pentagons and 0 heptagons. If you were having to have 13 pentagons, you need an heptagon to cancel it. This is a theorem. And it doesn't matter if it's a spherical or another shape, if it's close, it has to have these 12 defects, these 12 pentagons. So now I'll tell you what the pentagons do. That's the one that leads to the buckle. If it was something that has a genus 1, which means 1 toroid, like a toroid, like that, has 1 handle, then doesn't need defects because 1 minus 1 is 0. So you can actually take a tube and fold it and cover it and it doesn't spike, right? You don't need the defects that are so dangerous. So if you have and this is something we've known from Caspar and Klug for viruses, that the spherical viruses have icosahedral symmetry. And the reason for that is because these 12 defects hate each other. These 12 defects cause these little points hate each other. So if they hate each other and you have a sphere, they have to be further away from each other. They are in an inscribed icosahedron. So the position of the defects is like if you have inside an icosahedron. Okay? So this shape here. So, and because these effects, I'm going to show you why they hate each other. You will see it very clearly in this example. If it was a planar disk, and you have a defect there, this pentagon here, then it will be equivalent. This is a discretization of a continuum idea. It will be equivalent if you go to a bar and you have a round thing where you put your glass, divide it into 6 pieces, and take 1/6 of it, and try to glue it back. It makes a cone, right? That is the buckling. That's what we call the buckling. If it was elastic, it will close. But if it's not too elastic, it would buckle, depending on how much bending rigidity has that structure. So this is what we call, when it grows, the radius, the strain grows like R square of this little disk until it buckles and releases the strain into this cone. So suppose you have 12 of those in the sphere, and you keep growing the sphere. So the strain is growing like R square. So the big viruses would like to buckle and release the strain simultaneously by releasing the defects, and the shape it comes out is the icosahedron. So that's why it's so ubiquitous. So this is kind of like a theorem, right? Because I show you it has doesn't depend on the molecules. It's not molecular. It's just like a continuum theory of mechanics. So that's very nice. I mean, it's actually very recently that they understood this. Even though Caspar and Klug, that characterize the viruses as icosahedron and so on, they only know that the spherical ones have the symmetry on icosahedron, but they didn't know it was going to buckle and when. So this is a mechanics problem. Okay. So what happens if you make the shells of different triangles, ones that are called blue and red, some that are more rigid than the other. So this is what I call heterogeneities. So when you have these microcompartments or organelles, it's known that if they are made of one protein, they will be spherical. But if you make them of many proteins, they take many different shapes. So there has to be another mechanism for assembling things that have more than one component. And so many organelles are made of many different proteins. And so the proteins might have different local structure and rigidity if they aggregate. What I'm going to show you is that they aggregate to release the strain. So that's where you find these strange shapes, and I'll show you the mechanism of forming them. So these are the ones I call heterogeneous shells that are made of more than one component. The theorems, though, or the things I'm going to show you are for a continuum model. So it's just mechanics. If you have something that has two, like if you take a lamine thing that can have one soft part and one rigid part, and you close it, and so if you want it to be, crystalline or elastic, then what shape will take. And this is what this is part of, is going to tell you. So we start, we have to couple the shape with these displacements of the two components. This is a discretization of the continuum. You can make a mesh and make triangles and calculate how much energy you cost to change the angle between them. And that's the bending. And then you can calculate how much energy you pay for stretching the spring elastic. So when you take this into account and you do in the computer, you search all the shapes that it takes and take the minimum, the one that has lower energy. That's what we're doing here. Okay, you poke out and you calculate the angles and see whether it bends or changes shape or not. So these elastic materials are also non-local interactions. So what happens on one part is felt by the other part. So they are cooperative things, okay? Because they are connected. It's only one molecule, right? As I told you, if it's a crystalline or gel, it's one molecule. So if you have two types, you can exchange them in a computer by an algorithm. You decide if you lower the energy and you keep lowering the energy, and like that you find the shape. So we start mixing two components. One that was, if it was flat, was going to be rigid, with one that if it was buccal already, because it's more easy to bend. So when you mix them into one shell, what happens? That's the question. You have these two. So we start with, I'll show you what happened to them. When you look at the equilibrium, this is what happened. If this is, when it's pure, the one that is rigid, and this is the one that is buccal when it's pure. But if you have something, so these are the only two shapes that you can have by these theorems in shells, spheres or this buckle, right? But if you add, if I start, say, putting in the rigid one some of the subcomponent, then it will naturally aggregate and form these lines when it wants to merge. And they want to merge to something that we call fractionate the structure. They want to make as many possible faces it can. But when it's very little percent of the easy to bend, they can't not come close together. But when they manage to join, then they bend there and they release the strain like that. So as opposed to buckling the defects, it actually aggregates materials that are compatible. I mean, I'm putting the blue that likes the red, right? And they still segregate to release the strain. So it's another mechanism. But it's a very clever one because, it gives you all of these shapes that are much lower symmetry. Like in nature, if you were like to deliver something to a person, what shape would you use? A sphere, right? But if you want this object to have more function, then the sphere is just very simple, homogeneous. So you have to have more function to recognize a shape or to concentrate one component that you have very little of. Then it's very clever because it's going to use these shapes, specific shapes or whatever, that allows to concentrate, have other properties in the bands and so on. So this is another extreme of the simulations we do, which is continuum mechanics, and I show you the other cases of the atoms and so on. But it matches, actually. I will show you an example that, if you do the right physics and so on for the phenomena, and it has the correct principles, they will match. You will converge the microscopic with the continuum to find the shapes. So this is some of their, so I told you in the beginning, the Platonic Solids. You can, of course, get the Platonic Solids. This is a little bit, not fair because we put by hand how much percentage to form that Platonic solid. But in principle, they could exist by this mechanism, but not if they were homogeneous. So we have all these icosahedrons I told you in the beginning, and then the most common ones are the irregular, this. And so the mechanism of buckling is very different. As I told you, the defects are there still because my system has fixed connectivity. We didn't really change the mesh. We just minimized and let it change shape. And the way it buckled was by something that we call concentrating the curvature. Concentrating along these lines, so it bent more, the mean curvature was very high there. So that's other physicists study, for example, how you crumple a piece of paper. If you take a piece of paper and do that, it has the same principle. You concentrate the curvature. So it's very nice when things can be explained from different fields in this phenomenon. So now this is the elastic theory. I haven't put the charges here. Now, if you combine charge with elasticity, then you get much more interesting things. Okay? So this is an example of what can happen when we mix the 3 plus and minus 1. As I told you in the beginning, I'm going to demonstrate these shapes by another mechanism. Then we find that these structures can form, and I will show you how we do them in the lab to find them and so on. But before I do that, I'm going to show you this example so you see what electrostatics, the capability of electrostatics. These are objects that are the head groups 3 plus and then head groups minus 1. And so what we show here in this example, and this was a paper that won a Commissariat prize by the National Academy, it's very interesting that if you take a sphere and you just put spheres that have positive and negative charges. Now, the interaction between charges is isotropic, it's 1/r in any direction, okay? The spheres that I chose have spherical shape too. So when we put them in the surface of the sphere, it happens that it has lower energy if you have something faceted, by the pure electrostatics. Okay, so by pure charge, they want to be faceted. So which is something very interesting. And the resulting object, this was an icosahedron, but this icosahedron does not have icosahedral symmetry. And so let me explain this way. If you have a cube, it has a cube symmetry. But now if you are playing dice and you put labels 1, 2, 3, 4, 5, 6, now it doesn't have any more cube symmetry. You broke the symmetry because you have the labels. When these icosahedra have charges, what we find is that they break rotational symmetry, and each triangle in the icosahedra is not equivalent. They change the distribution. So it was very interesting because it means that this electrostatic interaction breaks symmetry spontaneously. And in order to have function, you have to break a symmetry. Okay? If you have something very symmetric, it's inert. So what I found it very, very striking is that it's only this 1/r potential, which is the one we're made of, charges, 1/r Coulomb and so on. So that's an example, just to tell you how you can build with charges. Now if you mix the two together, elastic and charges, then you can make kind of like little switches. So for example, if electrostatic is low, so the charges are in a medium where they are like in water, as opposed to say in oily when they are more strong, the interactions, because the electric media is lower. So they will form, the charges will be located in the positions of, if there are 8 charges in this case, will be a cube rotated. Okay? So the positions is a cube like that rotated. But if you increase electrostatics, it will go and make a cube not rotated. So it will have some sort of like switch 2 states. So that's just an example to show you when you are coupling more complicated interactions, the things you can do. And so now I'm going to design the things in the experiment and show you the shapes that we get and the changes they can go when you change what we call the ionic strength, the pH of the solution. So these they can dissociate one charge or the 3 of charges, and this one can dissociate -1. These molecules, this doesn't go into water because it's so hydrophobic, and this one forms the micelles. But if you just mix them and move them, then you will form, instead of just these micelles, you will form these buccal structures, these shells. Okay? So these are the shells that I was discussing, that they form in the beginning. But what we find surprising is that these shells form only at certain concentration of pH. pH is the concentration of H+, associated with the concentration of H+, that is in a solution. So depending on the amount of them, you can dissociate more charge to increase the concentration of them or not. So when you have very low pH, it means that some of the molecules are less charged. At this pH 7, both the positive and negative want to be both very charged. And at this other pH, the other molecule is not very charged. So the fraction of charge that you have in your system changes as a function of the pH. Mm-hmm. So what we notice is that at low pH, they have these closed shells, and at high pH. And at intermediate pH, they form ribbons, like sheets of material. So what I was trying to explain you with the pH gives you the fraction of the associated charge of the molecules. So here, the blues are the positively charged, and the red are the negatively charged. When you mix them, you can see that as a pH 3, only like around 30% of these are charged, and 70% are not charged. Okay? And so this tells you the fraction at the different pH of the blue and the red. So we assemble them, for example, in this case, at pH 3. So 70% of the ones that had -1 are uncharged. I call them here they're in color green. No, I'm sorry, gray. There's no green there, sorry. And the ones that are charged are the blue and red, the 3+ and -1. So when they are 30%, they are a bilayer like in the beginning I show you. In the other normal bilayers that we see in the membranes, right? But in a crystalline phase, though, in the phase where you have multivalent ions crystallized in them or low temperature. But if they have a pH 8, where you have 95% of them charged, then they kind of like overlap like that and collapse the layer. And so basically one can explain that, when you have some fraction without charge, this can sort of go in between. And when you have the more charge, they have to lower these sort of big head groups, charges, and overlap. So what is the relevance of that? So What happened physically to this is that the tails were arranged in a different symmetry than the hexagonal. Okay? So if you have a pH 3 here in all these regions, and here pH large, small and large, the tails have the symmetry of hexagons that they can rotate like that. But this was done by X-ray. We could look at exactly how they are packed, the tails. And in this region, they have like they are distorted. They are rectangular structures. They are called rectangular C. They are not anymore hexagons. So remember in the beginning, it was very important for all these theorems that you have these systems that come any shape, but they are always hexagonal prepacked. So to the conclusion we came from this study is that when you have not hexagonal packing in the layers, then they cannot close. Okay? They open up into the ribbons of these sheets. But when they have the hexagonal structure, they're able to close. And so when we go back to this model, which this is called a Martini model, that is a coarse grain with the 4 waters, 1 bead, and so on, then you can see that it looks very much like the full atom when we have all the water and the structures, the bilayer. And here they look disordered, right? So here there is more permeation, the properties are different, and in this respected by what I showed you in the beginning, with the properties of the bilayers. So the interesting thing is that maybe the reason why this can close only in hexagonal is because in here they are more disordered. They cannot pack in crystalline. And so when you have something in a disordered state, the normal number of neighbors is 6, right? And in a hexagonal is 6 nearest neighbors, as I showed you in the beginning for 1. So they have the same sort of numbers of neighbors, and they can, what we say, coexist, live in the same system, the 2 faces. If you have a system that is hexagonal, it cannot coexist in the same system with a rectangular C. So they cannot live together. They don't have a coexistence, is what we call it in material science, the term. So probably that's why they broken up. And so, but the interesting thing is I show you the continuum model that has nothing to do with the molecular model shows very similar results. If we just do, for example, assume that here is thinner than this part. So this one is more rigid, just by continuum mechanics, than these parts. And then we put those parameters in the modeling, and you get very similar shapes. Okay? So this is just, how everything will converge to the same sort of principle. But we predicted this by the continuum, and then we went back and designed the structures and showed that it does work. So this is a nice exercise, what you can do with soft matter and the tools we have now, computation and theory and so on. So, and of course, the experimentalists that are willing to go and try this in the labs, because it was all done theoretically. So here are the ribbons, that they break. And I'm just going to close with show you the summary and the reason why we believe in these rectangular C ribbons you cannot form closed structures. I'll show you very quickly. What will happen is that rectangular C looks more like a square lattice, right, as opposed to the other one. And so if you have a square lattice, why do you go from closed and triangular to square open? And this is a simulation where you start with a big sort of like drop of water, say, and you put, or oil in water, and you put charges there, just, fictitiously. And they are 1-to-1, so they have to form a square lattice. In a 1-to-1, you have to have all the nearest neighbors of the blue have to be red. So they have to be square. And then you start deleting the water, so it gets more and more concentrated. So if you do that, you go smaller, 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 and at some point, it has to form a hexagonal. So it not anymore can support a square lattice. So when it's dense packed, these structures do not support in the spherical shape the hexagonal structure. So you might think that that's why you don't make, the ceramics are ionic structures, plus minus. You don't find many things, they crack very easily and so on. The reason from a materials point of view is because the theorem I told you of the defects have to also obey here. But the defects are different. If you have a square lattice, how many neighbors you have? 4, right? So defects can have 3 neighbors or 5. okay? And they have to be close. But if they are ionic and they can have some stoichiometry of neighbors of charge, it's going to be very difficult to form these defects. This one is with 4. This is 4 and this is 5. 1, 2, 3, 4, 5. And, it looks like it's reasonably ordered, it can occur. The 3, which is the minimal one, has to be 3. Have 3 neighbors. They are very difficult to form. Generally, to be something plastic that has defects, the defects glide and behave nicely. But when you have charges, how do you put the defect that you don't violate from a red having a blue around? It's impossible, right? What is the line of defects you put here? You can't. In order to have, because if you put here, say, red, you are violating the red. And if you put blue, you are violating that. So in order to have a defect, you have to pair with another more complicated defect. And so it's very difficult to actually close these things in a sphere. And this is just, again, from another point of view. So it matches the results we find. So you can't. So I'm going to summarize saying that, we find these regular and irregular polyhedral geometries, and they arise spontaneously. What it means is that they buckle spontaneously. There's no force, They buckle spontaneously, and when they are formed more than one component. And these ionic crystalline membranes, which are ubiquitous in biology, have very unique functionalities, or we expect, such as the capability to change shapes. I show you some in response to external parameters, like the pH, and so on. We expect that in biology, the forces are similar to the ones I discussed here in orders of magnitude, so they are able to change shapes. Like if you have an ionic bonding, it's 100 kT. You don't change that, right, in a ceramic. But if it's 10 kTs, 5 kTs, and thermal energy is 1 kT, you can actually undergo many processes, and that's why we're so functional and can change and be soft and so on. So, and the ionic crystalline shells are stable at high salts and at high salt concentration, so they actually can survive in harsh environments. Interesting is they survive at high salts, but not by changing the pH, which is a different functionality. And so our models were able to predict this faceting, and I show you that these decorated polyhedra arise by the continuum model and also by the molecular models in the system. So with that, I'm just going to thank you, and sort of here are like all of the methods we use to describe this system that I told you in the beginning I was going to concentrate on. And I thank you. Okay. There is a question there. Oh.
Are we going to have a test, and do you grade on a curve?
I do grade on a curve, actually. So, but the lady on your... the pH.
Does this aid in coming up with vaccinations for viruses?
Yes, actually, people make the capsids only, without the RNA or the DNA, which is the one that is infectious, just so that your body can make antibodies recognizing some of the proteins and so on. But I don't work in applications like that. I'm just working the principles of what are leading to the shapes and so on. I mean, it'd be important to know how to design it, but to be honest, people work more, with trial and error in the specific proteins and so on. These are general principles. I show it with very simple amphiphiles, as opposed to the complicated proteins that are making the capsids. So here she, and then.
So, hello. Can you hear me? Okay. In Earth, I try to think of this in nature as well. So Earth is a sphere. Have you thought about where the defects would be in Earth?
That's a very good question. If it's solid, it doesn't need defects. It's only for shells. So because we have a core and so on. So it has to be membranes. This is for thin membranes. That's why it's for the bilayers only and for the nanocontainers. Yeah. So yeah, but if you have a single crystal, if there was a single crystal like the one I showed in the beginning that we make with the DNA and so on, or or your ring gems, then if it's single crystal, it has to show certain particular planes of atoms. And that's why they have these regular facet shapes, that they could look like Archimedean solids. But it's a different mechanism. That's called wolf-shaped construction. It's constructed just by minimum surface energy. The ones I show here is by buckling, and it's much more complicated because, I was changing the shape, the forming, and so on. Yeah.
I couldn't help but notice who funds your research and Department of Defense. Are there weapon applications for?
No, it is the only grant I had from the Department of Defense. Well, actually, I had a second one, but I was not the PI of that one. Was the National Security Science and Engineering Faculty Fellow, which is an award, was extremely generous by the basic science research of the Department of Defense. So it basically, the head of all of the Air Force and Naval and all of the other ones doesn't have budget himself, but he's the head of the basic science, and he has these fellowships. Now they changed name, but when I got it, they are like 10 a year, and they were like 30 right now. There will be like 40 National Security Science and Engineering Fellows, and so it was not for applications; it was for basic science. Very generous grant.
So I noticed that the intermediate stages between that spherical shape and the buckled stage there, I was just wondering if you've studied any other structures in nature that are naturally occurring, maybe like diatomic structures like that. I've just noticed that a lot of diatoms have that similar shape with the rounded, but then also that buckled form as well. It's like that in-between.
Right.
Have you maybe noticed that elsewhere or studied any of those naturally occurring, since they're larger and maybe not as small as like a virus?
Right. So these, as I said, they are membranes, very thin membranes. And so suppose if the material blue I put loves to be with the blue and hates the red, eventually it will concentrate. And then it will form a disk inside the red, and it will have different mean curvature because it bends differently. So it will have a sharp, some shapes like that, flat and so on. And they look more like, some, the atomic molecules you said, proteins. Yeah, but it's different mechanism that the proteins that have these complex shapes that can be sort of like cuboids and so on, they are just aggregates of proteins or distribution of how they form very structured proteins. They are more complicated actually because they have many amino acids to know exactly what shape they shape and so on. This, as I said, it was just a 2-component to show the mechanism, and it's only for shells. So it's not for bulk systems. They are shallow inside. They have to have water or.
Yes, so given our current battery technology that we're using with regular electrolytes that are non-renewable, what actions need to be taken?
Actually, I don't see who, oh yeah, sorry.
Yeah, sorry. Yeah, what action needs to be taken to transition to a lower cost, less volatile and recyclable alternative, such as polymer electrolytes?
We have to design the difficulty of the polymer electrolyte now is, for example, one of them is that you have to have the lithium moving. So because the electric media is very low, in terms of interactions, it means that if two ions in water interact until they come together at 7 angstroms with 1 kT. If you bring two ions in an electrolyte at that distance, they interact like with 70 kT. So that tells you the order of magnitude when there's no water. So it means that all of the electrostatic interactions are very large. So if you have a backbone of polymer and the lithium, it's so attached that it's very difficult to move it, to conduct. So what they try to do is make the charge groups on the backbone of the polymer very large, so that contact is not so close, so it's not so strong, and then when you can charge them and discharge them. But there are ways that people are doing it now. You can actually buy polymer electrolyte batteries made of polymers now. And so they are rechargeable too. Yeah.
Within your studies, did you notice that a specific pH of the shell had a highest functionality? And if so, what pH balance was that?
It's a kinetic process a little bit. The ones that have lower symmetry are the ones that are more functional. Now, by the X-ray, I say in the beginning that we know the shapes, we also know what is the size of the crystalline domain. And so if you take plates like that, and you have to close them into these sort of objects so that they don't have water details, if you make a small one, it's very faceted. Now if you tile with the same size of crystal, some very big sphere, it's spherical, right? Looks more homogeneous. So we find that the small ones were the ones that were more asymmetric. And so, the idea that we wanted to make something small that was like 50 nanometers, or 80 nanometers, we didn't want to make some very large structures. So we did find that the small ones have these sort of like more irregular shapes that the lower the symmetry, you will think the more functional, because the more, something different they can do. It's like if in the equivalent of functionality, suppose you have sticks, and then you pack them, and they're all equivalent all. You just make a hexagonal packing in this direction, but they're inert, right? You cannot, they are dense and so on. So if you take some shapes that are more decorated and so on, then they can recognize most things. So the lower the symmetry it has, the higher the functionality, generally.
Let's thank our speaker.